Hello, and welcome to another gentle journey on Sleepy Professor Stories. Tonight, you are invited to look at vegetables in a slightly different way. Not as items arranged on a plate, not as ingredients waiting to be prepared, but as quiet living systems. Imagine roots resting in cool, dark soil, feeling their way through tiny pockets of moisture. Picture leaves turning patiently toward the light, opening themselves each morning without hurry. Think of seeds lying still, sometimes for weeks or even months, waiting for just the right combination of warmth and water before they begin to stir. In this slow exploration, you will travel through ancient fields where early farmers gently selected wild plants and guided them season by season into the vegetables we know today. You will move from underground roots to tender stems, from unfolding leaves to delicate blossoms. You will notice how plant biology is not rushed or loud, but steady and rhythmic, shaped by sunlight, soil, and time. And as these living forms grow, you may begin to sense how their quiet work eventually becomes nourishment for the human body. If you find comfort in these calm journeys through science, you are welcome to like or subscribe in whatever way feels natural. It simply helps others discover a peaceful corner of learning too. But there is no pressure, no urgency. For now, there is nothing you need to do, nowhere you need to be. Let your breathing slow, adjust a little. Feel your body soften against whatever supports you. And if sleep arrives before we finish, that is perfectly fine. The garden will continue growing without you needing to listen to every word. Let's begin. You begin beneath the surface, where the world feels cool and dim, and the soil presses softly on every side. In this quiet place, seeds rest without urgency. They are not asleep in the way animals sleep, yet they are paused, held in a state of patient waiting. Inside, each one lives a tiny, complete plant, folded so carefully that it seems almost impossible. A root tip no thicker than a thread, a pale stem curved like a question mark, and one or two first leaves wrapped in protective layers. All of it is sealed within a coat that shields the life inside from dryness, cold, and time itself. Some seeds remain in this suspended state for months, even years, depending on the species and the environment around them. Their outer coats are designed to resist decay and prevent premature growth. This resting condition is known as dormancy, and it allows vegetables to survive winters, droughts, and long journeys in the wind or soil. Dormancy is not laziness. It is strategy. It is the plant's way of listening carefully to the world before committing to growth. When the soil warms and moisture seeps gently through the seed coat, something subtle begins to change. Water enters first, a quiet swelling as dry tissues drink and soften. This process, called imbibition, awakens the chemistry inside. Enzymes that have been inactive begin to stir. Stored food, packed away in the seed as starches, oils, or proteins, starts to break down into usable energy. You can imagine it as a lantern being lit in a dark room, not with a sudden flash, but with a gradual glow. Temperature plays its own quiet role. Many vegetable seeds germinate only within certain ranges of warmth. Cool season plants prefer the mild temperatures of early spring or autumn. Others wait for the deeper warmth of late spring. The seed does not think, yet it responds precisely. Within its cells, chemical reactions proceed more quickly when conditions are just right, guiding the transformation from stillness to growth. As water continues to enter, pressure builds inside the seed. The coat softens further, and at last, a tiny crack appears. The first structure to emerge is almost always the root. This initial root, called the radicle, pushes downward, guided by gravity. Even in complete darkness, it senses direction through specialized cells that respond to the pull of the earth. You might picture it gently feeling its way, anchoring the seedling in place and beginning to absorb more water and dissolved minerals from the surrounding soil. Soon after, the young shoot begins its own journey upward. It curves delicately, protecting the tender tip as it moves through the grains of soil. For some vegetables, the first leaves remain below ground, while others lift them into the light. These early leaves, known as cotyledons, are not true leaves in the full sense, but they serve as the plant's first source of nourishment, and sometimes its first green surfaces for photosynthesis. In that moment, when the shoot breaks through the soil and meets the air, a new stage of life begins, though it has been unfolding quietly all along. Light now becomes essential. While the seed relied on stored energy at first, the young plant gradually shifts to making its own food. In the presence of sunlight, chlorophyll develops within the cells, giving the leaves their soft green color. Through photosynthesis, carbon dioxide from the air combines with water drawn up by the roots, forming sugars that fuel further growth. What began as a dormant speck in darkness becomes a small, self-sustaining organism, turning light into life. And the soil itself is more than a passive backdrop. It contains countless microorganisms, bacteria and fungi that interact with emerging roots. Some form beneficial partnerships, helping the plant absorb nutrients such as nitrogen and phosphorus more efficiently. Others decompose organic matter, releasing minerals in forms that roots can take in. Even at this earliest stage, the seedling is not alone. It grows within a living community, each organism playing a subtle role. Moisture must remain balanced. Too little water and the fragile tissues dry out before they can establish themselves. Too much and oxygen levels in the soil decrease, suffocating the roots. Germination is a delicate negotiation between the seed and its environment. And yet, it happens quietly in gardens, fields, and forests across the world without announcement. As you imagine this unfolding, there is nothing you need to manage or remember. The seed knows its sequence. The soil holds its steady embrace. You can simply observe the rhythm of it all, the swelling, the softening, the first small push downward, the gentle rise upward. Beneath every vegetable plant you have ever seen lies this hidden beginning. Carrots, beans, lettuce, and pumpkins all pass through this silent threshold. They start as dormant seeds, carrying within them a blueprint shaped by generations of adaptation and careful cultivation by humans. Long ago, early farmers selected seeds from plants with desirable traits: larger roots, sweeter fruits, sturdier stems. Over centuries, this selection gradually shaped wild species into the vegetables that fill modern gardens. Yet even after thousands of years of cultivation, the essential process of germination remains unchanged. In the dark soil, the seed responds only to conditions: warmth, water, oxygen. It does not hurry. It does not compare itself to neighboring seeds. Each one begins when it is ready, when the signals align. Some sprout quickly within days. Others take longer, unfolding at their own pace. Growth is not a race beneath the earth. You might notice how fitting it is that life often begins in darkness. The absence of light is not an obstacle here, but a cradle. Protected from harsh sunlight and drying winds, the seed is given space to transform. Only when it has established a small root system does it rise to meet the brightness above. And so, in this quiet underground world, the story of every vegetable begins. In stillness, in patience, in a careful response to the environment. From a hardened shell to a living shoot, from stored reserves to active photosynthesis, from dormancy to growth. As you rest with this image of seeds gently awakening, you may sense how much depends on what lies unseen. Before leaves spread wide and stems stretch tall, there is always this hidden phase, this careful listening to the soil. And as we lift our attention upward from the darkness below, you may begin to wonder how those first tender roots continue their silent work once they have anchored themselves in the earth. Beneath your feet, in the cool and patient dark, roots extend themselves with quiet determination. They do not rush. They do not wander aimlessly. Instead, they grow with a kind of steady listening, responding to forces you cannot see, but that are always present. Gravity gently guides them downward, an invisible pull that roots sense through specialized cells near their tips. These cells contain tiny, dense structures that settle in response to gravity, helping the root understand which way is down. And so, even in complete darkness, a carrot root knows how to aim itself deeper into the earth. As the root tip moves forward, it is protected by a small cap of cells that shield its most delicate tissues from abrasion. This root cap secretes a slippery substance that eases its passage between soil particles, allowing it to glide rather than tear. Behind this protective tip, cells divide and then elongate, pushing the root slowly forward. Growth happens not in sudden leaps, but through steady cellular expansion, as if the plant is quietly unfolding itself into the spaces available. Water becomes one of the root's primary guides. Roots grow toward areas where moisture is more abundant, a response known as hydrotropism. In drier patches of soil, growth slows. In slightly damp regions, it continues with more confidence. The root is not aware in the way you are aware, yet it reacts precisely to gradients of water, adjusting its direction in subtle arcs. Over time, this responsiveness allows the plant to anchor itself in places where survival is more likely. Minerals dissolved in the soil also shape the journey. Nitrogen, phosphorus, potassium, and trace elements are absorbed through the root's outer cells. Tiny root hairs, almost invisible to the eye, extend outward from the main root body, dramatically increasing the surface area available for absorption. If you imagine the root as a hand, these hairs are like fine threads extending from each fingertip, gently touching the soil and drawing in nourishment. It is through these delicate structures that a beet gathers the elements it will later store in its rounded, ruby-colored body. Carrots, radishes, and beets are often called root vegetables, though what you eat is usually a modified taproot thickened with stored sugars. In their early stages, these plants send down a primary root that becomes the central axis of growth. As the plant above ground begins to photosynthesize, sugars produced in the leaves travel downward through specialized tissues, and accumulate in the root. Over time, this storage causes the root to swell. The carrot becomes firm and elongated, the radish crisp and compact, the beet dense and richly pigmented. What appears simple at the surface is the result of an intricate internal exchange between leaf and root. Roots also form branching networks. From the primary root, lateral roots emerge, spreading outward to explore a wider volume of soil. This branching pattern allows the plant to access nutrients and water beyond the immediate area beneath its stem. In some vegetables, fibrous root systems dominate, forming a dense web near the soil surface. In others, the central taproot remains the main anchor. Each pattern reflects adaptation to different environments and strategies for survival. The soil itself is alive with microscopic organisms. Certain bacteria attach to roots and assist in converting atmospheric nitrogen into forms plants can use, particularly in legumes. Fungi form partnerships, known as mycorrhizae, connecting to roots and extending far beyond their natural reach. Is a quiet collaboration. Unfolding is a quiet collaboration, unfolding beneath awareness, sustaining growth above. Oxygen, too, is essential below ground. Roots require oxygen for cellular respiration, the process by which stored sugars are converted into usable energy. If soil becomes waterlogged and air spaces fill with water, roots may struggle. This is why well-drained soil supports healthier vegetable growth. Even in darkness, even underground, the need for balance continues. Moisture, air, and nutrients must exist together in harmony. As you picture these underground movements, you may notice how gentle they are. Nothing forces the root forward. It responds to gradients, to slight differences in pressure and chemistry. There is no urgency in its growth. It advances millimeter by millimeter, adjusting as needed. You do not need to track its progress. It happens whether you watch or not. In cultivated fields, farmers prepare soil carefully, loosening it to allow roots to penetrate more easily. Compacted soil can restrict expansion, causing roots to twist or fork. When soil is soft and well-structured, roots grow straighter and more evenly. This is why a well-grown carrot is smooth and tapered, while one grown in rocky soil may bend around obstacles. More on the shape of the vegetable reflects the environment it experienced underground. Over time, the storage root becomes not only an anchor, but a reservoir. During cooler months or periods of stress, the plant can draw upon the sugars stored below ground. In some species, this storage allows the plant to survive winter and resume growth when temperatures rise. The root, hidden and silent, becomes a bank of energy, safeguarding the plant's future. You can rest with this image of roots listening to their surroundings. There is nothing you need to fix, nothing to calculate. The soil holds them gently. The earth's gravity guides them. Moisture invites them. Minerals nourish them. In their quiet way, they respond to all of it. Above the surface, leaves may tremble in the wind or bask in sunlight. But below, the roots remain steady. They form the unseen foundation of every vegetable plant, supporting the vibrant growth you eventually harvest. And as those roots continue to draw water and nutrients upward, you may begin to wonder how that invisible flow travels through the stem, rising quietly toward the leaves. You rise gently from the darkness of the soil into open air, where leaves unfold like quiet hands greeting the morning. They are soft, thin, and often delicate, yet within their fragile surfaces, unfolds one of the most important processes on Earth. In every green vegetable leaf, whether it belongs to spinach, lettuce, or cabbage, sunlight is welcomed and transformed. What seems simple at first glance is in fact a finely tuned system working steadily without sound. Each leaf is shaped to capture light. Broad and flat, it spreads outward to increase its surface area, positioning itself where sunlight is most available. In crowded gardens, leaves even adjust their angles, tilting slightly to avoid shading one another. Their green color comes from chlorophyll, a pigment nestled within tiny structures inside each cell. Chlorophyll absorbs specific wavelengths of light, especially red and blue, while reflecting green, which is why the leaf appears the color it does to your eyes. Inside those cells, in compartments called chloroplasts, sunlight sets a quiet sequence in motion. Water drawn upward from the roots enters the leaf through veins that branch like delicate rivers. Carbon dioxide drifts in from the air through microscopic openings called stomata. These pores open and close in response to light, humidity, and internal signals, balancing the need for gas exchange with the risk of losing too much water. Even here, there is careful regulation, a steady attention to balance. When light energy strikes chlorophyll, it excites electrons, initiating a chain of reactions that convert light into chemical energy. This energy is used to combine carbon dioxide and water into glucose, a simple sugar. Glucose becomes the building block for starch, cellulose, and countless other compounds that give structure and substance to the plant. Oxygen is released as a gentle byproduct, drifting back into the air you breathe. In this way, the leaf participates quietly in a global exchange, sustaining not only itself, but much of life on the planet. Spinach leaves, tender and slightly crinkled, are rich in chloroplasts, maximizing their capacity to harvest light. Lettuce forms layered rosettes, each leaf positioned to gather sunlight efficiently while protecting the younger leaves at the center. Cabbage wraps its leaves tightly around a growing core, yet even within those folded layers, photosynthesis continues wherever light can reach. Each vegetable expresses the same fundamental process through its own form. Veins run through every leaf, carrying water upward and transporting sugars downward. And the pattern of these veins strengthens the leaf and ensures that nutrients are distributed evenly. In many vegetables, the network is so fine that it resembles lace when held to the light. These veins are composed of specialized tissues. Xylem carries water and dissolved minerals from the roots, while phloem transports the sugars produced in photosynthesis to other parts of the plant. What begins as sunlight becomes nourishment stored in roots, stems, or fruits. Temperature influences this delicate work. Photosynthesis proceeds most efficiently within certain ranges of warmth. Too cold and the chemical reactions slow. Too hot and enzymes may lose their shape, reducing efficiency. This is why leafy vegetables often thrive in mild seasons, when sunlight is steady but not harsh. In extreme heat, stomata may close to conserve water, limiting carbon dioxide intake and slowing sugar production. Even in the act of gathering light, moderation remains essential. Water plays a dual role. It is both a raw material for photosynthesis and a cooling agent. As water evaporates from leaf surfaces through transpiration, it helps regulate temperature, preventing overheating under bright sun. This evaporation also creates a gentle pull that draws more water upward from the roots, maintaining a continuous flow. The leaf is not isolated. It remains connected to the entire plant through this circulating stream. You may imagine the leaf as a quiet kitchen where light is the fuel and carbon dioxide and water are the ingredients. But unlike any human kitchen, it operates without noise or haste. There is no visible flame, no scent of cooking, only the steady conversion of energy from one form to another. This transformation sustains the plant's growth and, indirectly, your own. Leaf structure also protects against excess light. Some vegetables produce pigments such as carotenoids, which absorb additional wavelengths and protect chlorophyll from damage. In red or purple varieties of cabbage and lettuce, anthocyanins contribute both color and protection. These pigments can shield cells from intense light and oxidative stress, allowing the plant to adapt to varying conditions. What appears decorative is often functional, a layer of resilience woven into beauty. As leaves mature, they adjust to their environment. In shaded conditions, they may grow larger and thinner to capture more light. In bright conditions, they often become smaller and thicker, reducing water loss. This flexibility reflects the plant's responsiveness to its surroundings. It does not resist change. It adapts quietly. There is nothing you need to hold on to here. The leaf continues its work whether you follow each detail or let your mind drift. Light arrives, water rises, carbon dioxide enters, sugars form, oxygen returns to the air. The cycle repeats countless times in every garden and field. When you look at a simple leaf of lettuce or spinach, you are seeing stored sunlight made tangible. The crisp texture, the subtle flavor, the nourishment it provides, all originate in this silent exchange between light and matter. The energy that began as photons traveling across space becomes chemical bonds within plant tissue. And when you eat these leaves, that energy continues its journey within you. As evening approaches and sunlight fades, photosynthesis slows and then pauses. Yet the leaf does not fall silent. It continues to respire, using stored sugars to maintain its cells. Even in darkness, life persists. The rhythm shifts, but it does not cease. You can rest in this steady pattern of light and transformation. Above the soil, leaves spread wide, drinking brightness and turning it into substance. And as that newly formed sugar travels downward through hidden channels, you may begin to wonder about the quiet pathways within the stem that carry these unseen rivers from leaf to root. You stand now in the quiet space between root and leaf, where the stem rises like a slender bridge. It may look simple from the outside, smooth or ridged, green or pale, yet inside it carries movement as steady as a slow river. Water drawn from the soil travels upward through delicate channels, while sugars made in the leaves drift downward to nourish roots and growing tips. Within every stalk of celery, every spear of asparagus, every branching head of broccoli, this circulation continues without sound. Inside the stem, tissues are arranged in long strands that extend from root to leaf. The xylem forms hollow tubes, strengthened by thickened walls. These tubes allow water and dissolved minerals to rise, pulled upward by the evaporation of moisture from leaf surfaces. As water escapes through tiny openings in the leaves, it creates a gentle tension that draws more water from below. This process, known as transpiration, is not forceful. It is a continuous, quiet pull, like a thread being drawn upward through fabric. You can imagine the stem as a column of microscopic vessels, each one contributing to the upward flow. In celery, these vessels are large enough to be seen as pale strings when you break the stalk. Those familiar threads are bundles of vascular tissue, the very pathways that once carried water to the leaves. Their crisp texture reflects the structural support they provide, helping the plant stand upright while conducting its hidden rivers. Alongside the xylem runs another tissue called phloem. Through this network, sugars produced in the leaves move toward areas that need energy or storage. Unlike the upward pull of water, sugar transport depends on differences in concentration. Where sugars are abundant, pressure builds slightly, and the solution flows toward regions where sugars are being used or stored. This movement can travel in different directions depending on the plant's needs. In a young broccoli plant, sugars may move toward developing buds. In asparagus, they may support the rapid elongation of tender shoots. The stem is not only a conduit, but also a support. Its outer layers provide strength, allowing the plant to resist wind and hold leaves in optimal positions for light capture. Fibers within the stem add resilience, while flexible tissues permit gentle bending rather than breaking. In broccoli, the thick stalk supports a cluster of immature flower buds, lifting them towards sunlight. In asparagus, the spear emerges from underground crowns and grows swiftly, relying on stored energy and efficient transport to reach upward. Water moving through the xylem carries more than moisture. It transports essential minerals absorbed by roots, including nitrogen, potassium, and magnesium. These elements contribute to processes such as protein synthesis and chlorophyll production. As water rises, it distributes these nutrients throughout the plant, ensuring that each leaf and growing point receives what it requires. The flow is continuous during daylight hours when transpiration is active, and it slows gently as evening approaches. Temperature and humidity influence this circulation. On warm, dry days, transpiration increases, pulling water upward more rapidly. On cool or humid days, the pull is gentler. The stem adjusts without conscious effort, responding to environmental conditions through the opening and closing of stomata in the leaves. This coordination maintains balance, preventing excessive water loss while sustaining nutrient delivery. You might picture these internal pathways as a living network, steady and reliable. There is no rushing within them, only a consistent rhythm shaped by sunlight, air, and soil. If you have ever placed a celery stalk in colored water and watched the tint climb upward, you have witnessed this movement made visible. The dye travels through xylem vessels, tracing the route water normally follows. Even in such a simple experiment, the plant's hidden architecture reveals itself. As plants mature, stems may thicken through the activity of cambium cells, which produce new vascular tissue. This growth strengthens the structure and expands its capacity for transport. In some vegetables, stems remain tender and herbaceous, while in others, they become more fibrous over time. Yet, in all cases, the same fundamental system operates, linking root and leaf in a seamless exchange. Oxygen and carbon dioxide also pass through stems, though most gas exchange occurs in leaves. Tiny openings in the stem surface, called lenticels, allow gases to diffuse in and out. Even in the supportive tissues, respiration continues. Cells consume sugars to generate energy, sustaining growth and repair. The stem, like every living part of the plant, remains active day and night. There is nothing you need to follow too closely here. The water rises, the sugars descend, the stem holds steady. You can let your thoughts drift like the currents within it, moving gently from one image to the next. The plant does not hurry its circulation, and neither must you. In asparagus fields at dawn, tender shoots push upward with remarkable speed, supported by efficient transport from root reserves. In broccoli patches, sturdy stalks lift clustered buds toward open air. In garden beds, celery stands crisp and upright, its channels quietly conducting moisture and minerals. Each example reflects the same principle: structure and flow intertwined. When evening settles and transpiration slows, the upward pull decreases. Yet internal adjustments continue. Sugars redistribute. Cells repair and prepare for another day of light. The stem remains the quiet mediator between darkness below and brightness above. As you rest with this image of hidden rivers moving through living columns, you may sense how everything within the plant is connected. Nothing grows alone. Roots draw water, leaves create sugars, and stems unite them in a balanced exchange. And as this internal rhythm continues, you may begin to wonder about the blossoms that sometimes rise at the tips of these stems, opening softly to the air and inviting another form of quiet transformation. You move now toward the tender tips of stems, where buds gather quietly, almost shyly, before they open. Many of the vegetables you know begin here, in blossoms that face the sky for only a short while. Before there is fruit, before there is seed, there is a flower, delicate, structured, purposeful. Cauliflower, artichoke, and squash all remind you that nourishment often begins in beauty, in petals that unfold softly in morning light. A flower is not decoration alone. It is the reproductive structure of the plant, carefully arranged to bring together pollen and ovules. Within its layered form are stamens that produce pollen and a central pistil that receives it. Pollen grains carry the male genetic material, light enough to be moved by wind or carried by insects. When pollen reaches the receptive surface of the pistil, a slender tube grows downward toward the ovary, allowing fertilization to occur. This process is quiet and precise, guided by chemical signals within the flower's tissues. In squash plants, bright yellow blossoms open wide, inviting pollinators such as bees. These flowers often bloom early in the day when pollinators are most active. Some blossoms produce only pollen, while others contain the ovary that will swell into fruit once fertilized. Without the transfer of pollen from one flower to another, the familiar squash would never form. You may picture the brief meeting between bee and blossom, a gentle exchange that sets the next stage of growth in motion. Cauliflower offers a different perspective. What you eat is not a fully opened flower, but a tightly packed cluster of immature flower buds. If left unharvested, the compact white head would elongate and bloom into small yellow flowers. Farmers harvest it before this transformation, capturing the stage just before full flowering. In this way, you are eating potential, a paused moment in the plant's reproductive cycle. Artichokes, too, are unopened flower buds. Their layered bracts protect a central cluster that, if allowed to mature, would reveal a striking purple bloom. The vegetable you recognize is a snapshot in time, a blossom gathered before it completes its natural unfolding. The tenderness you taste reflects this early stage, when tissues are still soft and concentrated with nutrients. Pollination can occur in different ways. Some vegetable flowers rely on insects drawn by color and scent. Others depend on wind to carry pollen between plants. The shape and arrangement of floral parts influence how effectively pollen is transferred. In many cases, flowers are structured to encourage cross-pollination, increasing genetic diversity. Yet some are capable of self-pollination, ensuring reproduction even when pollinators are scarce. Inside the fertilized ovary, seeds begin to develop. Hormones trigger the surrounding tissues to expand, forming fruit that protects and nourishes the seeds within. In squash, the ovary swells into the familiar shape that ripens over weeks. In peppers and tomatoes, similar processes occur, though their flowers may appear smaller and less showy. What seems like a simple vegetable is often the matured result of a successful floral union. Temperature and daylight influence flowering. Many plants respond to the length of day, a phenomenon known as photoperiodism. Certain vegetables initiate flowering only when days reach a specific duration. This timing ensures that pollination and seed development occur during favorable seasons. The plant senses changes in light through pigments that measure day length, adjusting its growth accordingly. Even without awareness, it aligns itself with the rhythm of the year. Nutrients also shape floral development. Adequate phosphorus supports energy transfer within cells, aiding the formation of buds. Balanced soil conditions contribute to sturdy stems that can hold blossoms upright. Stress, whether from drought or nutrient deficiency, may reduce flowering. Yet, when conditions are supportive, buds emerge with quiet reliability. You do not need to track each detail. The flower opens in its own time. Pollen drifts or is carried gently from one bloom to another. Fertilization occurs within hidden chambers. Seeds begin to form, cradled inside protective tissues. All of it unfolds without noise. As blossoms age, petals may wither and fall, their work complete. What remains is the swelling ovary or the developing cluster that will become harvest. This transition from flower to fruit or bud is subtle but profound. It marks the shift from invitation to protection, from openness to enclosure. In gardens, the presence of flowers signals continuity. They are not only beginnings, but bridges between generations. Through them, genetic information passes forward, allowing vegetables to adapt and persist. The cycle repeats season after season, linking past and future in a steady rhythm. You may notice how gentle this process feels. There is no rush in the opening of petals, no urgency in the transfer of pollen. The breeze carries grains lightly. Insects move from blossom to blossom without intention beyond their own needs. Yet together, these small actions sustain entire fields of growth. As evening approaches, many flowers close, conserving energy and protecting delicate structures. Others remain open into twilight, awaiting nocturnal pollinators. The cycle of opening and closing follows patterns shaped by light and temperature. Even in rest, preparation continues. You can rest here as well. There is nothing you need to influence. Flowers have been opening and closing long before you noticed them. They will continue long after. Their brief beauty supports lasting nourishment, and their quiet labor ensures the next stage of life. As petals fall and developing fruits begin to take form, you may begin to wonder how some of these ripening structures blur the line between flower and vegetable, inviting a closer look at what we truly mean when we name what we harvest. You stand now in a place of gentle reconsideration, where familiar names soften and shift. The tomato resting in your kitchen, the pepper glowing red or gold, or the cucumber cool and green. These are often called vegetables in daily speech, yet in botanical language they are fruits. This is not a contradiction, only a difference in perspective. Science looks at structure and origin, while cooking looks at flavor and use. Both views can exist side by side, like two paths through the same garden. In botanical terms, a fruit is the mature ovary of a flower. After pollination and fertilization, the ovary begins to swell. Its walls thicken, now forming layers that protect the developing seeds inside. The seeds themselves carry the next generation of the plant, each one holding a tiny embryo and stored nutrients. The surrounding flesh is not random. It forms to shield, nourish, and sometimes entice animals to carry those seeds elsewhere. When you slice open a tomato, you see chambers filled with seeds suspended in gel-like pulp. Those chambers are the transformed sections of the flower's ovary. The outer skin develops from the ovary wall, becoming firm enough to protect the contents, but thin enough to allow ripening. As the tomato matures, pigments shift from green to red, orange, or yellow, signaling readiness. Chlorophyll fades, and other pigments become visible. What began as a blossom now holds seeds wrapped in soft tissue. Peppers follow a similar pattern. Their hollow interiors cradle seeds attached to a central core. The bright color that appears as they ripen is not merely aesthetic. It reflects chemical changes within the fruit, often increasing sugar content and altering flavor. These changes can attract animals, encouraging them to eat the fruit and disperse the seeds. Even in cultivated gardens, this evolutionary strategy remains quietly present. Cucumbers develop from flowers with visible ovaries at their base. After successful pollination, the ovary elongates, forming the familiar cylindrical shape. Inside, seeds arrange themselves along a moist center. The crisp flesh surrounding them serves both as protection and as an appealing texture for potential dispersers. In nature, animals that consume the fruit may carry seeds far from the parent plant, depositing them in new soil. From a culinary perspective, these foods are grouped with vegetables because of their savory flavors and use in meals. Botany, however, relies on structure rather than taste. If it develops from the ovary of a flower and contains seeds, it fits the definition of fruit. This distinction reminds you that language adapts to context. Scientific classification aims for clarity about origin and function, while everyday language reflects cultural habits. Some fruits we call vegetables are technically berries. In botanical terms, a berry forms from a single flower with one ovary and has seeds embedded in flesh. Tomatoes and peppers meet this definition. Strawberries do not, none because their seeds sit on the outside and the edible portion develops from other floral parts. Such details reveal how classification depends on precise criteria rather than appearance alone. The layers of a fruit often serve distinct roles. The outer layer, sometimes called the exocarp, forms the skin. The middle layer, the mesocarp, becomes the fleshy portion you eat. The inner layer, the endocarp, surrounds the seeds. In tomatoes and cucumbers, these layers blend softly together. In other fruits, like peaches, the inner layer hardens into a stone. Though you may not think of these vegetables as fruits, their anatomy follows the same blueprint. Ripening involves coordinated chemical processes. Enzymes break down cell walls, softening the texture. Sugars accumulate or become more noticeable as starches convert. Pigments shift and aromas develop. These changes are regulated by plant hormones, particularly ethylene in many species. Ethylene acts as a signal, promoting ripening in the fruit itself and sometimes in nearby fruits as well. Even after harvest, this hormone continues its quiet influence. There is no need to memorize the terms or trace every pathway. You can simply observe how perspective shapes understanding. The same tomato can be a vegetable in your salad and a fruit in a botanical text. The same pepper can be both garnish and seed-bearing structure. Science invites you to look beneath familiar names and see how form and function intertwine. The seeds enclosed within these fleshy walls are not passive. Each seed holds genetic information shaped by generations of selection, both natural and human. Farmers have long chosen fruits with desirable traits: larger size, sweeter flavor, thicker flesh, guiding the evolution of these plants over centuries. Yet, despite this guidance, the fundamental structure remains. A flower becomes an ovary. The ovary becomes a fruit. The fruit protects seeds. In warm sunlight, fruits develop color and flavor gradually. Temperature, water availability, and nutrient levels influence their growth. Too little water may limit size. Too much may dilute flavor. Balanced conditions allow sugars and acids to reach harmonious proportions. Even in cultivated fields, environmental rhythms shape each harvest. You might imagine the fruit as a quiet cradle, holding seeds safely until the moment of dispersal. In wild landscapes, animals eat fruits and carry seeds elsewhere. In gardens, humans harvest and plant seeds intentionally. In both cases, the fruit serves its primary purpose, ensuring the continuation of life. There is nothing you need to sort or resolve here. Words shift gently. Categories overlap. The plant continues its cycle regardless of what you call it. Flower, fruit, vegetable. These are lenses through which you view the same living structure. As you rest with this understanding, you may feel how perspective can expand without conflict. The tomato remains what it is, whether named fruit or vegetable. The cucumber continues to ripen in sunlight. The pepper glows quietly on the vine. And as these seed-filled forms complete their development, you may begin to wonder about the remarkable ways plants store energy below the surface, preparing for seasons yet to come. You return now to the quiet darkness beneath the soil, where certain vegetables do not simply anchor themselves, but transform into living storehouses. Potatoes and sweet potatoes rest here, thick and unassuming, holding within them energy gathered from long days of sunlight. They are not roots in the strictest sense, though they grow underground. A potato is a modified stem, a swollen structure that forms at the end of an underground branch. A sweet potato, by contrast, is an enlarged root. Both serve a similar purpose. They store nourishment for the plant's future. Imagine the process unfolding slowly across a season. Leaves above ground gather light and convert it into sugars. Some of these sugars fuel immediate growth, building stems and expanding leaves. Others travel downward through phloem tissues, arriving at underground organs prepared to store them. There, sugars are transformed into starch, a compact form of energy that can remain stable over time. And the plant is not only living in the present, it is quietly preparing for what lies ahead. In a potato plant, slender underground stems called stolons extend outward from the main plant. At their tips, certain cells begin to swell as starch accumulates. This swelling becomes the tuber. What you recognize as the potato is a collection of stem tissue filled with stored carbohydrates. The small indentations on its surface, often called eyes, are actually buds. Each eye holds the potential to produce a new shoot under the right conditions. If placed in moist soil and given warmth, those buds awaken and send up stems toward the light. Sweet potatoes form differently. They develop from specialized roots that thicken as starch builds within them. Unlike potatoes, their surface lacks visible buds, and new plants are typically grown from slips, which are shoots that emerge from the stored root. Though their origins differ, both structures embody the same strategy: to gather energy when it is abundant and conserve it when conditions grow uncertain. Storage organs allow plants to survive through periods of cold or drought. In temperate climates, winter can halt growth above ground. Leaves wither and stems may die back. Yet beneath the soil, tubers and storage roots remain insulated by earth. Their stored starch sustains vital tissues until warmth returns. When spring arrives, new shoots draw upon these reserves, pushing upward before leaves have had time to gather fresh energy. This early start offers an advantage, allowing the plant to reclaim sunlight quickly. You can picture the tuber as a quiet pantry. Sunlight once captured by leaves now rests in dense, pale flesh. The warmth of summer has been condensed into granules of starch. Even after harvest, these structures remain metabolically alive, though slowly. They respire gently, or consuming small amounts of stored energy to maintain cellular integrity. Proper storage conditions, cool, dark, and well ventilated, help slow this respiration, preserving the tuber's resources. Inside a potato, cells are packed with starch granules, each one a compact cluster of glucose molecules. These granules are insoluble in cold water, allowing the tuber to hold large quantities of energy without disrupting cellular balance. When cooked, heat breaks down cell walls and gelatinizes starch, making it more accessible for digestion. What was once the plant's reserve becomes nourishment for you. Hormones guide the formation and dormancy of tubers. During active growth, certain signals encourage the development of stolons and the swelling of their tips. As daylight shortens or environmental conditions shift, other hormonal changes promote dormancy, preventing premature sprouting. This balance ensures that stored energy is conserved until growth is favorable. Sweet potatoes, with their orange flesh, also contain pigments such as beta-carotene, which serve protective roles in plant tissues. These compounds can act as antioxidants, helping the plant manage cellular stress. When you eat them, those same pigments contribute to your nutrition, linking the plant's internal strategies with your own biological needs. There is something profoundly patient about these underground stores. They do not display themselves. They wait. In darkness, insulated by soil, they hold energy quietly. The plant's above-ground life may fade with the season, yet below, reserves endure. You do not need to hurry this understanding. The plant has already done its preparation. Leaves gathered light, stems transported sugars, and cells arranged starch into stable forms. The tuber rests, complete in its purpose. In agricultural fields, farmers often harvest tubers before the plant fully declines, carefully lifting them from the soil. The act of harvest interrupts the plant's cycle, but preserves the stored energy for human use. Through cultivation, humans have selected varieties with larger storage organs, smoother textures, and richer flavors. Yet the underlying biological strategy remains ancient. Even in wild ecosystems, similar storage adaptations exist. Many perennial plants develop bulbs, corms, or rhizomes to survive adverse seasons. The principle is consistent. Capture energy when it is plentiful, store it safely, and draw upon it when needed. It is a lesson in foresight embedded in living tissue. As you rest with this image of tubers nestled beneath the earth, you may sense the quiet assurance they represent. Energy once scattered as sunlight has been gathered and held. Growth pauses, but it is not forgotten. Beneath still soil, possibility waits. And as these hidden reserves support new shoots in the returning light, you may begin to wonder how human hands have guided such cycles across centuries, shaping wild plants into the familiar companions of gardens and fields. You step now into a longer stretch of time, far beyond a single growing season. Imagine early landscapes where wild plants grew freely, their seeds scattered by wind and animals, their flavors often sharp or bitter, their roots slender and unpredictable. Long before organized farms or garden beds, humans began to notice which plants were nourishing, which were easier to gather. Many vegetables looked very different from what you know today. Early carrots were thin and pale, sometimes purple or white rather than orange. Wild cabbages grew along coastal cliffs with loose leaves and small buds. The first forms of corn bore tiny ears, each holding only a few kernels. Through repeated gathering and planting of seeds from plants with desirable traits, humans gently guided change. This process, now called domestication, unfolded over generations, not through sudden transformation, but through patient selection. When early farmers chose seeds from plants with larger roots or milder leaves, they were influencing genetic variation without understanding the underlying mechanisms. Within every population of plants, natural variation exists. As some individuals produce slightly bigger fruits, others resist pests or tolerate drought more effectively. By saving seeds from those plants and replanting them, humans increase the frequency of these favorable traits. Over time, the cumulative effect became visible. Vegetables grew larger, sweeter, or more tender. This quiet collaboration did not happen in a single place. In different regions of the world, people cultivated distinct species suited to local climates. In parts of Asia, leafy greens and soybeans were carefully tended. In the Americas, beans, squash, and maize were shaped by attentive hands. In Europe and the Mediterranean, cabbages and root crops were gradually refined. Each culture contributed to the shared story of cultivation, adapting plants to soil, weather, and dietary needs. Selective breeding operates through inheritance. Traits are passed from parent plants to offspring through genes carried in seeds. When humans consistently select certain traits, those genes become more common in subsequent generations. This method differs from modern genetic modification, which involves direct alteration of DNA in controlled settings. Traditional breeding relies on natural variation and cross-pollination, guided by careful choice rather than laboratory tools. Over centuries, domesticated vegetables often became less bitter and more palatable. Bitterness in wild plants frequently serves as a defense against herbivores. By favoring milder varieties, humans reduced concentrations of certain defensive compounds. At the same time, they increased sugar content or starch storage, enhancing caloric value. The balance between protection and flavor shifted gradually under human influence. Color also changed through selection. The familiar orange carrot emerged relatively recently in history, likely favored for its vibrant hue and sweetness. Purple and white varieties still exist, reminders of earlier forms. Similarly, different shades of peppers and tomatoes reflect both natural diversity and deliberate breeding choices. Pigments that once served protective functions became markers of preference and identity. As agriculture developed, techniques improved. Farmers learned to rotate crops, enriching soil by alternating species with different nutrient needs. They discovered that legumes could restore nitrogen to depleted fields through partnerships with soil bacteria. Irrigation systems allowed cultivation in drier regions, expanding the range of possible crops. These practices reflected growing understanding of ecological balance, even before scientific terminology described it. Domestication often reduced a plant's ability to survive without human care. Many modern vegetables rely on cultivated environments, prepared soil, and protection from competition. Wild ancestors were often more resilient in harsh conditions. Yet, in exchange for this dependence, domesticated plants produced greater yields and more predictable harvests. The partnership deepened, each side adapting to the other. Seeds became precious carriers of lineage. Families saved seeds from favored plants, passing them down through generations. Over time, distinct varieties emerged, each suited to particular climates or culinary traditions. These heirloom varieties preserve genetic diversity, offering resilience against changing conditions. Diversity within crops remains important, as it allows adaptation to pests, diseases, and shifting weather patterns. In more recent centuries, scientific understanding of genetics refined breeding practices. Knowledge of inheritance patterns allowed more deliberate crossing of varieties to combine desired traits. Yet even with advanced tools, the fundamental principle remains unchanged. Select, plant, observe, and repeat. The rhythm of cultivation continues to follow seasons and patience. There is no need to hurry through this history. It unfolds slowly, like the growth of a field from scattered seeds to organized rows. Humans and vegetables have shaped each other quietly. Through nourishment, plants sustained communities. Through care and selection, people guided plant evolution. You might imagine ancient hands pressing seeds into soil, unaware that their choices would echo through millennia. Each saved seed carried not only genetic information, but also cultural memory. The flavors you taste today reflect countless small decisions made long ago. In modern gardens, this partnership persists. Gardeners choose varieties based on climate, taste, and resilience. Farmers experiment with new crosses, seeking resistance to emerging diseases. Researchers conserve seed banks to protect diversity for the future. The relationship remains dynamic, responsive to both environmental and societal change. You do not need to trace every lineage or remember every origin. The vegetables around you already embody this shared history. Their shapes, colors, and flavors tell a story of gradual transformation, guided by attentive observation. As you rest with this sense of continuity, you may notice how cultivation reflects both patience and foresight. Seeds saved today shape harvests tomorrow. Choices made in one season ripple into the next. And as this long partnership between human and plant continues, you may begin to wonder how the subtle chemistry within leaves and roots gives rise to the colors that catch your eye in a garden at dusk. You step into the garden at a softer hour, when sunlight feels gentle and colors seem to glow from within. Orange carrots resting beneath feathery greens, purple eggplants hanging like polished lanterns, deep green kale leaves curling at their edges. Color in vegetables is not decoration alone. It is chemistry made visible, a quiet expression of how plants gather light, protect themselves, and adapt to the world around them. The most familiar color in any garden is green. This shade comes from chlorophyll, the pigment that captures sunlight and allows photosynthesis to unfold. Chlorophyll absorbs red and blue wavelengths of light while reflecting green, which is why leaves appear the color they do. Within tiny compartments inside each plant cell, chlorophyll molecules sit ready to catch passing photons. Without them, leaves would not transform light into sugars, and growth would slow into stillness. Yet green is only the beginning. In carrots and sweet potatoes, the bright orange hue comes from carotenoids. These pigments absorb excess light energy and help protect plant tissues from damage. When sunlight is intense, chlorophyll can become overwhelmed. Carotenoids act as quiet guardians, dissipating surplus energy and preventing harm. They also contribute to the plant's resilience under stress, now helping maintain cellular stability. What you see as vibrant color is also a shield. In eggplants and red cabbage, purples and deep reds arise from anthocyanins. These pigments can change shade depending on acidity within plant cells, appearing more red in acidic conditions and more blue in alkaline ones. Anthocyanins often accumulate in response to environmental challenges, such as strong light, cold temperatures, or nutrient imbalances. By absorbing certain wavelengths and reducing oxidative stress, they protect delicate tissues. Their beauty is functional, not accidental. Yellow peppers and golden squash reflect additional carotenoid varieties, each contributing subtle differences in hue. As fruits ripen, chlorophyll breaks down and other pigments become visible. This shift often signals readiness for seed dispersal. In wild settings, bright colors attract animals that eat the fruit and carry seeds elsewhere. In cultivated gardens, the same signal guides human harvest. Even white vegetables carry color in hidden forms. Cauliflower appears pale because its developing flower head is shielded from light by surrounding leaves. Without exposure to sunlight, chlorophyll does not accumulate. If those leaves are pulled back, the curd may turn green as chlorophyll forms. The absence of color tells its own story about light and growth. Pigments also influence flavor and nutrition. Carotenoids can be precursors to vitamin A in human diets. Anthocyanins are associated with antioxidant properties. While plants produce these compounds primarily for their own protection and function, They become part of the nourishment you receive. Chemistry within plant cells connects quietly to processes within your body. Light intensity shapes pigment production. Under stronger sunlight, some vegetables deepen in color as protective compounds increase. Cooler temperatures can enhance anthocyanin accumulation, giving kale or cabbage a richer tone. The environment leaves its imprint on color, just as it shapes size and texture. No two seasons produce exactly the same shades. You might imagine pigments as subtle filters placed over a window. They allow certain wavelengths to pass while absorbing others. Through this selective capture, plants regulate how much energy enters their systems. Balance remains essential. Too little light limits photosynthesis. Too much can damage cells. Pigments help maintain equilibrium. In leaves, chlorophyll is often accompanied by accessory pigments that broaden the range of light absorbed. These companions capture wavelengths that chlorophyll misses and transfer that energy inward. Together, they maximize efficiency. Beneath what appears to be a single shade of green lies a complex arrangement of molecules working in harmony. As autumn approaches in some regions, the chlorophyll breaks down more quickly than other pigments. Carotenoids and anthocyanins become visible in leaves that once seemed purely green. While many vegetables are harvested before such changes occur, the principle remains. Color reflects internal shifts in chemistry. There is nothing you need to memorize here. You can simply notice how color becomes a language. Green speaks of photosynthesis. Orange whispers of stored light and protection. Purple suggests resilience under challenge. Yellow hints at ripeness and readiness. When you hold a carrot freshly pulled from the soil, its brightness carries traces of sunlight converted and stored. When you slice an eggplant, its glossy skin reflects pigments that once shielded living tissues from excess light. Each hue tells a story of adaptation. Color also varies within a single species. Through selective breeding, humans have encouraged diversity in shades and patterns. Heirloom tomatoes display stripes, marbling, and deep crimsons. Peppers range from pale ivory to almost black. These variations arise from differences in pigment concentration and distribution. Genetics guides these patterns, but environment influences their expression. As evening settles, colors soften, yet pigments remain present within cells. Even in darkness, their molecular structures persist, ready to resume work with the return of light. The garden rests, but chemistry continues quietly. You can rest as well, letting the images of orange, purple, and green drift gently through your thoughts. The colors do not demand attention. They simply exist, reflecting careful balances inside living tissues. And as you linger with these shades in fading light, you may begin to wonder how the same chemistry that creates color also shapes the flavors that unfold when you taste a leaf or bite into a fruit. You pause now with a simple question resting softly in your mind. Why does one leaf taste sweet, while another carries a gentle bitterness? Why does a freshly sliced cucumber feel cool and mild, while a radish offers a quiet heat? Flavor, like color, is chemistry made tangible. Within the tissues of every vegetable, compounds form that shape what you taste and smell. These molecules serve the plant first, yet they also create the experience you recognize as flavor. Sweetness often begins with sugars formed through photosynthesis. Glucose and fructose circulate within plant tissues, are sometimes stored as starch, sometimes remaining dissolved in sap. In vegetables like carrots and peas, higher sugar content creates a mild, pleasing sweetness. During growth, sugar levels can fluctuate depending on sunlight, temperature, and water availability. Cool nights sometimes slow respiration, allowing sugars to accumulate and deepen flavor. Bitterness arises from different compounds. In leafy greens such as kale or mustard, glucosinolates contribute a sharp edge. These molecules contain sulfur and can break down into pungent substances when plant cells are damaged, such as during chewing. This reaction evolved as a defense, discouraging herbivores from consuming too much of the plant. What you perceive as bitterness is part of a protective strategy. Radishes and other members of the same botanical family share similar chemistry. When you bite into a radish, enzymes mix with stored compounds, producing isothiocyanates that create a peppery sensation. Now, the plant does not aim to challenge you. It simply carries a built-in response to injury. In moderate amounts, these compounds can add complexity to flavor, while also playing roles in plant health. Aromas come from volatile molecules that evaporate easily and travel through the air to your nose. In herbs like basil or parsley, essential oils accumulate in specialized cells. When leaves are crushed, these oils are released, filling the air with scent. Now, these aromatic compounds can attract pollinators in flowering plants or deter pests that might otherwise feed on leaves. Their fragrance is both invitation and defense. Onions and garlic contain sulfur-containing compounds that produce sharp aromas when cut. Inside intact cells, these chemicals remain separated. Once tissue is damaged, enzymes combine them, forming new molecules responsible for their familiar scent. This transformation happens in moments, illustrating how flavor often depends on interaction between previously isolated substances. Texture also shapes flavor perception. The crispness of a fresh carrot reflects the integrity of its cell walls and water content. As vegetables age or cook, cell walls soften and moisture redistributes, altering how flavors are experienced. Heat can break down starch into simpler sugars, increasing sweetness. It can also reduce bitterness by altering chemical structures. The taste you notice changes as chemistry shifts. Some flavors develop during ripening. As fruits like tomatoes mature, acids and sugars reach a balance that defines their characteristic taste. Early in development, higher acidity may dominate. As ripening progresses, sugar levels rise and certain volatile compounds increase, contributing to aroma. Ethylene, a plant hormone, often regulates this process, coordinating changes within the fruit. Environmental conditions influence flavor chemistry. You shift the balance between sugars and shift the balance between sugars and acids. Each harvest carries traces of the season in which it grew. You might think of flavor as a conversation between plant and environment. The plant produces compounds in response to sunlight, soil nutrients, and potential threats. You encounter these compounds when you eat the vegetable. Your taste buds detect sweetness, bitterness, saltiness, sourness, and umami through specialized receptors. Signals travel to your brain, where sensation becomes experience. There is no need to analyze every molecule. You can simply notice how sweetness feels gentle, how bitterness can be grounding, how aroma awakens memory. Plants evolved these chemicals for survival, yet they also enrich your meals and connect you to the garden's subtle complexity. Fermentation offers another layer of transformation. When vegetables such as cabbage are fermented, microorganisms convert sugars into acids, creating tangy flavors. This process preserves the food while introducing new compounds. The chemistry shifts again, guided by microbial activity rather than plant enzymes. Even after harvest, flavor continues to evolve. You may find it comforting that bitterness and sweetness coexist within the same leaf. Balance defines much of what you enjoy. Too much sugar can overwhelm. Too much bitterness can deter. Together, they create nuance. Plants manage this balance through metabolic pathways that adjust to internal and external cues. There is nothing you need to solve here. The chemistry unfolds whether you study it or not. Sugars form in sunlight. Defensive compounds accumulate in leaves. Aromatic oils gather in tiny glands. When you slice, chew, or cook, these compounds meet air and heat, revealing their character. In fields and gardens, this chemistry operates quietly every day. Farmers and gardeners may select varieties known for sweetness or mildness, shaping flavor through breeding. Yet the core processes remain rooted in plant biology, guided by enzymes and environmental signals. As you rest with these gentle layers of taste and scent, you might sense how flavor tells a story of protection, attraction, and adaptation. The same molecules that defend a leaf can nourish you. The same sugars that fuel growth can satisfy hunger. And as you let these sensations drift softly through your thoughts, you may begin to wonder how the rhythm of changing seasons shapes not only flavor, but the very timing of growth itself. You notice now how no vegetable grows in isolation from time. Each one responds to the slow turning of the earth, to the tilt of its axis, to the lengthening and shortening of days. Seasons do not announce themselves loudly to plants. Instead, subtle shifts in temperature, light, and moisture guide quiet adjustments within stems and leaves. Growth and rest follow a rhythm as steady as breathing. Some vegetables prefer the cool embrace of spring and autumn. Spinach, lettuce, peas, and broccoli thrive when air remains mild and soil carries gentle warmth without heat. In cooler conditions, now certain chemical processes unfold more efficiently. Leaves remain tender, and sugars can accumulate without being rapidly consumed by respiration. If temperatures climb too high, these same plants may bolt, sending up flowering stems prematurely in response to stress. Bolting reflects an attempt to complete reproduction before conditions worsen. Warm season crops, such as tomatoes, peppers, and squash, require deeper warmth to flourish. Their seeds germinate best when soil temperatures rise beyond early spring chill. Enzymes within their cells function optimally within a warmer range, allowing steady growth. If frost touches their tissues, damage can occur as ice crystals form within cells and disrupt delicate membranes. Frost is not dramatic in its arrival. It settles quietly, yet its impact can be profound. Day length plays an equally important role. Some vegetables respond to photoperiod, and the duration of light within each 24-hour cycle. Onions, for example, form bulbs only when days reach a specific length. Short-day varieties begin bulbing when daylight shortens, while long-day types wait for extended sunlight. The plant senses these changes through pigments that measure light exposure, adjusting growth patterns accordingly. Even without awareness, the plant aligns itself with the calendar of the sky. Soil temperature influences root activity. In early spring, when soil remains cool despite warming air, root growth may proceed slowly. Nutrient uptake depends on root metabolism, which is sensitive to temperature. As soil warms, roots become more active, absorbing water and minerals more efficiently. You might picture the soil as a slow conductor, warming gradually and signaling when it is time to expand. Water availability shifts with seasons as well. Spring rains may saturate soil, while summer heat draws moisture upward and away. Vegetables adapt through adjustments in leaf size, root depth, and stomatal behavior. In dry conditions, stomata close partially to reduce water loss, though this also limits carbon dioxide intake and slows photosynthesis. Balance remains central, even under changing skies. Perennial vegetables and related plants often use dormancy to survive unfavorable periods. During winter, metabolic activity decreases. Leaves may die back, while underground organs conserve energy. Hormonal signals maintain this resting state until warmth and light return. Dormancy is not failure. It is preparation. Stored reserves support regrowth when conditions improve. You may find comfort in how consistently plants respond to seasonal cues. There is no panic in their adaptation. When days shorten, growth slows. When frost approaches, tissues harden slightly, accumulating solutes that lower the freezing point of cellular fluids. This process, known as cold acclimation, enhances survival in cooler climates. The plant adjusts quietly, molecule by molecule. Gardeners often work with these rhythms rather than against them. Cool season crops are planted early, harvested before intense heat arrives. Warm season varieties wait until soil warms sufficiently. Crop calendars reflect generations of observation, aligning human activity with natural cycles. Timing becomes a partnership between intention and environment. Sunlight angle also changes with seasons, influencing intensity and duration of exposure. In summer, higher sun angles provide stronger light, accelerating growth for suitable crops. In winter, lower angles reduce intensity, slowing photosynthesis even on bright days. Vegetables respond by adjusting leaf orientation and internal processes, optimizing available energy. There is nothing you need to calculate here. The Earth tilts gently, orbiting the Sun in a steady path. Temperature rises and falls. Daylight expands and contracts. Vegetables respond without hesitation, guided by internal chemistry attuned to external signals. As autumn settles, some vegetables concentrate sugars in their tissues, improving flavor after light frosts. Cold can convert stored starch into sugars, making roots such as carrots taste sweeter. This shift is another adaptation, helping protect cells from freezing damage while subtly altering taste. In spring, seeds sense warming soil and increasing moisture, initiating germination. Shoots emerge in synchrony with longer days, ready to capture sunlight. The cycle resumes, not as repetition, but as renewal. You might imagine the garden as a quiet clock, each plant marking time in its own way. Leaves unfurl, flowers bloom, fruits ripen, then rest returns. No season lingers forever. Growth gives way to pause, and pause prepares for growth. As you rest with this gentle cycle, you may sense how everything unfolds at its proper pace. Nothing forces the seasons forward. Nothing rushes them back. Vegetables adapt, endure, and thrive within these rhythms. And as you consider this steady turning of the year, you may begin to wonder about the unseen connections beneath the soil, where roots and microorganisms continue their quiet exchanges regardless of the season above. Beneath the surface, where light does not reach and sound seems to fade, a different kind of exchange continues. You have already felt how roots extend downward, searching for water and minerals. Yet they do not explore alone. Around every root tip lives a community of organisms too small to see without magnification. Bacteria drift through films of moisture between soil particles. Fungal threads weave through the earth like delicate silk. Together, they create a living network that hums quietly beneath your feet. When a young root begins to grow, it releases small amounts of sugars and other compounds into the surrounding soil. This release, called exudation, is not waste. It serves as a signal and an invitation. Microorganisms respond to these compounds, gathering near the root surface. Some of these microbes assist in breaking down organic matter, freeing nutrients that the plant can then absorb. In this way, the root feeds the soil community, and the soil community feeds the root. Among the most remarkable partnerships are those formed with fungi, known as mycorrhizae. The word means fungus root, and it describes a close association between fungal filaments and plant roots. The fungus extends fine threads, called hyphae, far beyond the reach of the plant's own root hairs. These threads explore pockets of soil that would otherwise remain inaccessible. In exchange for sugars produced through photosynthesis, the fungus delivers minerals such as phosphorus and sometimes nitrogen back to the plant. You might imagine this as a quiet trade agreement in darkness. The plant offers energy in the form of carbohydrates. The fungus offers access to distant resources. Neither speaks, yet chemical signals guide their cooperation. Structures form where fungal threads penetrate or wrap around root cells, creating intimate points of exchange. These connections do not harm the plant. Instead, they enhance its ability to thrive in nutrient-limited soils. Bacteria also play important roles. Certain species live freely in the soil, decomposing fallen leaves and organic debris. Others associate more closely with roots. In legumes, specialized bacteria can inhabit root nodules, converting atmospheric nitrogen into forms usable by the plant. While many common vegetables are not legumes, they still benefit from microbial activity that cycles nutrients through the ecosystem. Nitrogen, once locked in complex molecules, becomes available again through microbial transformation. Communication within this underground world often relies on chemical signals. When a root encounters stress, such as drought or pathogen attack, it may alter the compounds it releases. Nearby microbes can respond, sometimes increasing protective activities. In some cases, signals can travel through interconnected fungal networks, linking different plants. Though it would be inaccurate to imagine conscious messaging, the exchange of chemical cues creates coordinated responses across the soil community. The structure of soil itself influences these interactions. Healthy soil contains aggregates of mineral particles bound together with organic matter and microbial secretions. These aggregates create tiny air pockets that allow oxygen to diffuse downward. Roots require oxygen for respiration, and fungi need it as well. Balanced soil structure supports both plant and microbial life, sustaining their quiet partnership. Even pathogens participate in this hidden dialogue. Some fungi and bacteria can cause disease when conditions favor their growth. Yet beneficial microbes often compete with or suppress harmful organisms, maintaining balance. Diversity within the soil community increases resilience. When many species coexist, no single one dominates completely. This ecological balance protects vegetable plants from widespread damage. Moisture weaves through all of this. This water forms thin films around soil particles, allowing nutrients to dissolve and move. It carries chemical signals between roots and microbes. Too little moisture limits microbial activity. Too much may reduce oxygen and disrupt delicate equilibria. Once again, balance shapes the health of the underground world. You do not need to follow every microscopic interaction. It is enough to picture a vast web beneath the garden, threads of fungi connecting roots, bacteria cycling nutrients, roots releasing gentle signals into the soil. Even in stillness, activity continues. As seasons change, these relationships shift subtly. Cooler temperatures may slow microbial metabolism. Warmer conditions can accelerate decomposition and nutrient release. Yet even during winter, many microorganisms persist, maintaining low levels of activity and preparing for spring's return. Gardeners sometimes encourage these partnerships by adding compost or organic matter to soil. Compost provides food for microbes, enriching the community that supports plant growth. Practices that avoid excessive chemical disturbance help preserve the intricate networks already present. Cultivation becomes not only the care of plants, but the care of soil life. There is something reassuring about this unseen cooperation. Vegetables do not stand alone, isolated in dirt. They are embedded in a dynamic system where exchange and support occur continuously. The roots that listen to gravity and water also listen to chemical whispers in the soil. You may find it calming to imagine these conversations unfolding quietly as you rest. No sound reaches your ears, yet communication persists. Sugars move outward, minerals move inward, signals pass between cells and filaments. In this darkness, cooperation sustains growth above ground. Leaves drink light, stems carry water, roots share and receive. The garden becomes more than individual plants. It becomes a network. As you linger with this image of subtle exchange beneath the surface, you might begin to wonder how the nutrients gathered through these hidden partnerships eventually find their way into your own body, linking soil and human in one continuous cycle. You bring the garden a little closer now, not just to your plate, but to your own quiet interior. The journey that began in dark soil and rose through stem and leaf continues within you. When you eat a carrot, a handful of spinach, or a spoonful of beans, the stored sunlight and gathered minerals enter a different landscape, one shaped by blood vessels, cells, and gentle chemical exchanges. Vegetables offer vitamins, small organic compounds that your body requires in modest amounts, yet cannot always produce on its own. Vitamin C, present in peppers and leafy greens, participates in the formation of collagen, a protein that supports skin, blood vessels, and connective tissue. It also acts as an antioxidant, helping neutralize reactive molecules that form during normal metabolism. When you consume these foods, vitamin C dissolves into your bloodstream, traveling quietly to where it is needed. Vitamin A often begins in vegetables as beta-carotene, a pigment stored in orange and dark green tissues. Inside your body, beta-carotene can be converted into active forms of vitamin A, which support vision, immune function, and cellular growth. The same molecules that once protected plant cells from excess light now assist your own cells in maintaining their structure and clarity. Minerals absorbed by roots find their way into your circulation as well. Iron, present in leafy greens and legumes, contributes to the formation of hemoglobin, the protein that carries oxygen through your blood. Magnesium participates in hundreds of enzymatic reactions, including those involved in muscle function and energy production. And potassium helps regulate fluid balance and supports steady heart rhythms. These elements do not act alone. They integrate into complex systems, maintaining balance with quiet precision. Fiber, abundant in vegetables, travels a different path. Unlike sugars and vitamins, fiber is not digested into simple molecules. Instead, it moves through your digestive tract largely intact. Soluble fiber can absorb water and form a gel-like substance, slowing digestion and helping regulate blood sugar levels. Insoluble fiber adds bulk, supporting regular movement through the intestines. In both forms, fiber creates an environment that nourishes beneficial gut bacteria. Your digestive system responds gently to this plant material. Chewing breaks cell walls, releasing nutrients and allowing digestive enzymes to access them. In the stomach and small intestine, acids and enzymes break down carbohydrates, proteins, and fats. Nutrients cross the intestinal lining into the bloodstream, while fiber continues onward, fostering microbial activity in the large intestine. This collaboration between plant compounds and human biology reflects a partnership shaped over evolutionary time. Antioxidants present in vegetables contribute to cellular protection. Compounds, such as flavonoids and carotenoids, can help neutralize free radicals, molecules that arise naturally during metabolism and in response to environmental stress. While your body has its own antioxidant systems, plant-derived compounds can complement these defenses. The colors that once shielded plant tissues from sunlight now participate in protecting your cells. Circulation carries absorbed nutrients throughout your body. Blood vessels form branching networks reminiscent of plant veins, delivering oxygen and nutrients to tissues and returning waste products for removal. In this way, the structure of plants echoes within you. Water, minerals, and sugars once moved upward through stems. Now, nutrients move through arteries and capillaries. There is no need to treat this knowledge as instruction. You do not have to calculate exact amounts or track every micronutrient. It is enough to recognize that vegetables contribute to ongoing maintenance and repair. Cells renew themselves continuously. Skin regenerates. Blood cells are replaced. Nutrients provide the raw materials for these quiet processes. The relationship between plant and person extends beyond chemistry. When you prepare vegetables, you engage in acts of care that connect soil to kitchen. Heat can alter nutrient availability, sometimes reducing certain vitamins while increasing accessibility of others. For example, cooking can soften cell walls, making some carotenoids easier to absorb. Balance again emerges as a theme. Raw and cooked forms each offer distinct advantages. Hydration also plays a role. Many vegetables contain significant amounts of water, contributing to overall fluid intake. This water supports circulation, temperature regulation, and metabolic reactions. The moisture that once moved through xylem vessels now participates in your own internal flows. Gut microbes interact with plant compounds in complex ways. Certain fibers are fermented by bacteria, producing short-chain fatty acids that can support intestinal health. These interactions illustrate another layer of partnership, linking plant chemistry to microbial ecosystems within you. You may notice how seamlessly these processes occur. There is no conscious effort required for absorption, circulation, or repair. Your body, like a plant, responds to available resources with coordinated adjustments. Nutrients enter, cells respond, balance is maintained. Rest with this awareness. The same sunlight that touched a leaf now supports your vision. The same minerals drawn from soil now participate in muscle contraction. And the same pigments that colored a carrot now contribute to immune defense. There is nothing urgent here, only continuity. Plants gather energy and nutrients. You consume them. Your body integrates what it needs and releases what it does not. The cycle continues. As you linger in this quiet exchange between garden and self, you may begin to wonder how preserving vegetables through drying or fermentation extends their nourishment across time, allowing stored sunlight to remain available even when fresh harvests fade. You stand now at the gentle edge between abundance and waiting. A garden has given generously, leaves and roots gathered in baskets, colors still bright with recent sunlight. Yet harvest is not only about eating in the present. It is also about carrying nourishment forward, extending the life of vegetables beyond their natural span. Preservation is patience made practical, a quiet agreement with time. A drying is one of the oldest methods. When moisture leaves plant tissue, microbial growth slows dramatically. Without water, bacteria and molds struggle to multiply. In thin slices of peppers or herbs laid out in warm, circulating air, water gradually evaporates. Cells collapse slightly as they lose moisture, concentrating sugars and flavors. The vegetable does not spoil, it transforms. Its life processes cease, yet its stored energy remains. Fermentation follows a different path. Instead of removing moisture, it invites selected microorganisms to reshape the vegetable's chemistry. When cabbage is shredded and mixed with salt, salt draws water from the cells, creating a brine. In this salty environment, certain bacteria naturally present on the leaves begin to thrive. These lactic acid bacteria convert sugars into lactic acid, lowering the pH and creating conditions inhospitable to harmful microbes. What emerges is sauerkraut, tangy and stable. The same principle applies to cucumbers submerged in brine to become pickles. In the absence of oxygen and with sufficient salt concentration, beneficial bacteria outcompete others. Over days or weeks, acids accumulate and flavor deepens. Texture shifts slightly as cell walls interact with salt and microbial enzymes. Preservation is not stagnation. It is transformation guided by balance. Temperature plays a quiet role in these processes. Cool storage slows enzymatic activity and respiration in fresh vegetables, extending their shelf life. Refrigeration reduces the rate at which cells consume stored sugars. In root cellars, stable cool temperatures and moderate humidity keep carrots and potatoes firm for months. The vegetable remains alive but rests, its metabolism subdued. Salt and acidity both function as guardians. Salt draws out moisture and inhibits unwanted microbial growth. Acidity creates an environment where pathogens struggle to survive. Vinegar pickling uses externally added acid, while fermentation generates acid internally through microbial action. Both methods rely on principles of chemistry and microbiology rather than force. You might imagine fermentation as a gentle conversation between plant and microbe. The vegetable offers sugars. Bacteria respond, producing acids and subtle flavor compounds. No rushing occurs. The transformation unfolds gradually, guided by temperature, salt concentration, and time. If conditions are balanced, the result is stable and nourishing. Enzymes within vegetables also change during storage. In some cases, starches continue to convert into sugars, subtly altering taste. In others, prolonged storage may reduce certain vitamin levels. Preservation does not freeze time completely. It slows it, redirecting chemical pathways rather than stopping them outright. Drying intensifies flavor because water, which once diluted sugars and acids, is removed. A dried tomato tastes richer than a fresh one because the same compounds now exist in a smaller volume. Sun drying uses warmth and airflow to evaporate moisture. Modern methods may employ controlled heat, but the principle remains unchanged. There is no urgency here. Vegetables that once grew in open fields now rest in jars, cellars, or woven baskets. Their forms shift, but their purpose continues. Preservation extends nourishment into seasons when fresh growth is limited. It bridges harvest and winter, abundance and quiet. Even freezing, a more modern technique, relies on slowing microbial and enzymatic activity. At low temperatures, water inside cells forms ice crystals. If done quickly, smaller crystals form, reducing structural damage. When thawed, the vegetable softens slightly, yet nutrients remain largely intact. The stored sunlight still offers energy. You do not need to measure every temperature or salt ratio in your thoughts. It is enough to sense how patience shapes preservation. Time becomes an ally rather than an adversary. Microbes are not enemies, but partners when guided appropriately. In many cultures, fermented vegetables have long supported communities through colder months. The tang of sauerkraut or kimchi reflects both plant chemistry and microbial artistry. These foods embody adaptation to climate and season, carrying harvest forward when gardens lie dormant. As you imagine rows of jars resting quietly on shelves, you may feel a sense of calm continuity. The vegetables have changed form, yet their origin remains in soil and sunlight. The cycle of growth does not end with harvest. It simply enters another phase. There is nothing you need to prepare or preserve right now. The transformation has already occurred in countless kitchens and cellars. You can simply rest with the image of cabbage slowly turning sour, cucumbers becoming crisp and tangy, peppers drying into concentrated sweetness. And as preserved vegetables wait patiently through winter's stillness, you may begin to wonder how, in the quiet of night, living plants themselves continue their subtle work, even after darkness settles over the garden. Evening settles gently over the garden, and the light that once poured across leaves begins to fade. Shadows lengthen. Colors soften. What seemed vibrant and busy in the afternoon now appears still. Yet beneath this calm surface, life continues. Plants do not sleep in the way animals do. They shift their chemistry, adjusting to darkness with quiet precision. During the day, leaves capture sunlight and convert it into sugars through photosynthesis. When darkness arrives, that particular process slows and then pauses because light is no longer available to drive it. But respiration continues. In every living plant cell, sugars are broken down with oxygen to release energy. This energy supports maintenance, repair, and growth. Even without sunlight, the plant remains active, sustaining itself from reserves gathered earlier. You can imagine respiration as a slow internal glow. Sugars formed during daylight are now gently consumed. Oxygen enters through tiny pores, diffusing into tissues. Carbon dioxide, produced as a byproduct, drifts back into the night air. This exchange is steady and unhurried. It does not require brightness to proceed. Stomata, the microscopic openings on leaf surfaces, often close more fully at night to conserve water. With cooler temperatures and higher humidity, water loss decreases. The plant conserves its resources while continuing cellular work. In some species, subtle movements occur. Leaves may fold slightly or change orientation, a behavior known as nyctinasty. These movements are regulated by internal clocks and shifts in water pressure within cells. Circadian rhythms guide much of this activity. Plants possess internal timing mechanisms that anticipate the cycle of day and night. Even if kept in constant light or darkness, certain processes continue to follow roughly 24-hour patterns for a time. These rhythms regulate gene expression, enzyme activity, and growth rates. The plant prepares for dawn before the first light appears. Roots remain active below ground. Water uptake may slow at night, but nutrient absorption can continue. Microbial partners in the soil persist in their own cycles, decomposing organic matter and releasing minerals. The underground network does not pause simply because the sky has darkened. Growth often occurs most noticeably in darkness. In some vegetables, cell elongation increases at night, allowing stems or leaves to extend slightly by morning. Without the demands of photosynthesis, our resources can be allocated towards structural expansion. The change may be subtle, measurable only with careful observation, yet it reflects continuous life. Temperature typically drops after sunset, influencing metabolic rates. Cooler conditions can slow respiration slightly, conserving stored sugars. In some climates, nighttime cold triggers protective responses, prompting the accumulation of solutes that help prevent cellular damage. Even in stillness, adjustment continues. You might feel reassured by this steady persistence. The garden does not depend on constant light to remain alive. It adapts, redistributing energy, maintaining tissues, preparing quietly for the next sunrise. Insects move differently at night as well, and some flowers open only after dusk to attract nocturnal pollinators. Though many vegetable crops complete pollination during daylight, the broader plant world reminds you that darkness carries its own forms of interaction. Moisture gathers on leaves as dew when air cools to its saturation point. This thin film of water can influence microbial activity on leaf surfaces. Yet plants are adapted to these cycles, balancing risk and resilience through evolved responses. You do not need to monitor each exchange of oxygen and carbon dioxide, each subtle shift in enzyme activity. The garden manages its own rhythms. You can simply imagine the leaves resting under starlight, roots nestled in soil, stems holding their quiet form. Respiration links plant and person in another way. Just as plants consume oxygen and release carbon dioxide at night, you do the same continuously. The air between you and the garden becomes a shared medium, molecules drifting gently through darkness. As midnight deepens, metabolic processes continue at low levels. Sugars are mobilized where needed. Cells repair minor damage from daylight exposure. Internal clocks count down toward morning. There is no urgency in this waiting. Dawn will arrive in its time. You may picture a row of cabbages under moonlight, their broad leaves silvered at the edges. Beneath the soil, carrots remain cool and still. In the quiet air, faint scents linger from herbs and earth. Nothing dramatic occurs, yet everything persists. There is comfort in knowing that life does not require constant attention. It flows through cycles of brightness and shadow, activity and rest. And the garden teaches this balance without words. As this journey gently comes to a close, notice how wide and still the world feels. So many quiet gardens growing in darkness, whether we see them or not. You don't need to remember every leaf or root. They have already done their work. If you enjoy these slow explorations, you can support the channel in any small way that feels natural, so others can find this calm too. But right now, there is nothing to decide and nowhere to go. Let your thoughts soften, your muscles relax, and the night carry you onward, one peaceful breath at a time.